science. Our next speaker is Dr. Hollmann now from the Ruhr University in Bochum, and he's giving a talk about a beautiful cooperation with Aslam. He just died, J. van Engelhardt, SF Trinellis, and SC Bering. And the title of his talk is, again, in Russian, please translated.
So difficult to translate from Russian to English here. Okay, now try to avoid any accents. Modulation of function of glutamate receptors by interactions with the membrane-associated proteins. You understand?
Very good, very good, very good.
Yeah, very good.
So somehow he knows quite some Russian. Okay, please go ahead. You're right on time, you have no hurry, no rush.
Yeah, well, really happy, that's news. No, it's a beer. And again, we don't see the people from the, let's say, local people who help us.
You don't need help.
Oh. We pretend that we cannot.
We will all remember this workshop. You see hundreds in a year, but we will remember. Yeah.
I would be happy to hear your remembrance after 100 years. Okay.
I'm not sure that I'm not alive this time.
Okay, I hope this stays working. Thanks a lot, Victor, for inviting me here. It's probably the most unusual session I've ever been in 25 years. So, but yeah, that's
but no complaints.
No complaints. I don't know why it's doing this. I hope it's not going to be a problem on all the slides. Anyway, I'm kind of a maverick here in this session because I'm going to show you that there's scientific life beyond cis-loop receptors. Because I'm working on glutamate receptors, and I have been ever since we found the first glutamate receptor back in 1989 in the Hollmann lab. And just to remind you, because you might not be that familiar with the glutamate receptors, there is quite a number of genes, 18 total subunits, and the AMPA receptors, 4 subunits, are responsible for fast excitatory neurotransmission, basal neurotransmission. The kainate receptors participate in this. There's 5 genes of those. Also have a presynaptic function in glutamate and agonist release. Then there's arguably the most important glutamate receptors, the NMDA receptors. There's 5 of those genes, mostly important for everything that concerns synaptic plasticity, so learning, memory formation, and such. And let's skip the delta receptors because they're not really well known yet. And this is the structure of the glutamate receptor as revealed by Eric Deveau a few years ago. And as you can see, there's 4 subunits, so not 5. And if you look at the structure of a single subunit, as you can see down here, is there a way to get this window off here? No, I don't think so. The structure of these single subunits are such that we have a large extracellular domain, 3 transmembrane domains, and a hairpin loop here, this green one, that actually forms the ion channel. So it's not a transmembrane domain forming the ion channel in this case, and a large C-terminal domain. And the field in glutamate receptors has moved now to a stage where it has been recognized that the receptors are not sitting all by themselves in the membrane and doing their thing, but they are intimately associated with a lot of proteins that modulate their function. So functional modulation by all kinds of proteins is important, and I just show you a whole slew of these proteins that have been discovered as interacting proteins. And the first ones were all directed to the C-terminal domain, the ones shown here in black. And later on, people discovered that there's actually proteins that interact in the plane of the membrane So they are themselves a transmembrane domain carrying proteins and are sort of sideways interacting with the glutamate receptor. And there's a few proteins, very few, that are acting only on the extracellular part. And I'll stick with the intramembranous ones in my talk. And the granddaddy of all of these are the TARPs. They were the first discovered transmembrane proteins that modulate glutamate receptors. And starting from the TARPs, we actually looked at homologs of TARPs and discovered a completely new family of interacting proteins that nobody thought about, that looked quite unusual actually to be true, the claudins, which turned out to contain some modulatory proteins as well. And just to introduce you to the TARPs were originally discovered in this mouse, which is called the stargazer mouse, because it always seems to look up at the stars, tosses its head in the back, and that's why they call it the stargazer mouse. And the protein responsible for that, that's mutated in this particular mouse, is called stargazing. And the function that was found, or the lack of function, was found in cerebellar granule cells, where people showed that in the homozygous mutant mouse, there's absolutely zero AMPA receptor TARPs. In the very same cells, cerebellar granule cells, however, you find nicely expressed NMDA receptor TARPs, so they are fine. It's just the AMPA receptors lacking in this. And when this was further analyzed, then people found that this molecule responsible for this phenotype and mutated in the stargazer mouse, called stargazin, is actually a member of the gamma subunit family of calcium channels. And these calcium channels, as you know, they have a lot of accessory proteins, and one is the gamma subunit. In muscle calcium channels, for example, gamma 1 modulates that channel. But in addition to gamma 1, there's 7 additional gamma subunits, all the way up to gamma 8. And stargazin is gamma 2. And this one modulates the glutamate receptor along with 6 other ones out of these 8 gamma subunits. 6 of them are actually modulators of AMPA receptors. Only 2 of them are only restricted to calcium channels. So then they were given a new name. They were termed TARPs, transmembrane AMPA receptor regulatory proteins. And they basically have 2 functions. They further the trafficking of AMPA receptors to the membrane. So they make sure you have enough AMPA receptors in the membrane, or in the case of the stargazer mouse, none. And they also modulate the function of the receptor. They are tetraspanins, so they are sitting in the membrane like this, 4 transmembrane domains, C and N termini intracellularly, and they sideways interact with the glutamate receptor. So the closest relative of this family of TARPs and gamma subunits happens to be the claudin family. Now claudins, all of you who are biochemists probably know what claudins are. Claudins are tight junction proteins. So they're well known to form tight junctions. So a tight junction is formed by having claudin molecules put like beads on a necklace in strings in the adjacent membranes of 2 adjacent cells, and then seal this up. And it is sealed up so well that not even a tracer molecule, a radioactive tracer molecule, if you put it up here, can penetrate down here beyond the tight junction. Or if you put the tracer down here, it will penetrate up through the tight junction in the upper space, if you have 2 epithelial cells next to each other with a tight junction in between. So the way this works is that the claudin molecules have 2 extracellular domains, and they connect with the extracellular domains from the claudin to the other cell, and then they tighten up this intercellular space. So there was one published observation by Prince et al. In 2005, where they actually showed that the TARPs have one property in common with the claudins. The claudins, as I told you, make the tight junction, so they are cell-cell adhesion furthering proteins. So if you express claudins in mouse fibroblasts, the cells start to aggregate. Here's the control. Normal fibroblasts don't aggregate. Now the same thing happens if you express a TARP in these mouse fibroblasts. They start to aggregate. Now we reason, okay, if TARPs can mimic a claudin function, as shown here, maybe claudins can also mimic the TARP function in modulating the glutamate receptor. And this is actually what happens. And now, for some reason, the data slide won't present here. That's too bad. Okay, so I have to explain this to you. The family of claudins is really big. There's 28 subunits. And we tested most of them. So a poor PhD student of mine had to subclone all of them to look at them, because we had no idea which one would be an active one. And it turns out that claudins 20 and 24, and of course he started with claudin 1. 20 and 24 turned out to have a potentiating effect on glutamate receptors. And well, sorry for that. That's the problem if the technique doesn't cooperate. We tested this on the GluA1 receptor first, so the AMPA receptor GluA1, and then tested this on other glutamate receptors, GluA2, GluA3, GluA4, and it turns out that claudin 20 is very specific just for GluA1. It doesn't touch GluA2 and GluA3, or GluA4 for that matter. Here's now. See, 24 and 20 and 24 were the ones that had this potentiating effect. And in the presence of a desensitization inhibitor, the effect still is there. So you get this potentiation of only these two receptors. And this is GluA2, 3, and 4. And you see for those, there is little, if any, potentiation. So specific for GluA1 receptor. Now, if we look at another receptor, which is a calcium impermeable glutamate receptor, which has an arginine at the core of the receptor of the ion channel. This arginine makes the receptor calcium impermeable. And when you look at these calcium impermeable receptors, then an interesting thing happens. So for all four of them, GluA1, 2, 3, and 4, claudin 21 all of a sudden becomes a strong potentiator. Your effect is up to 40 times more. But only for this calcium permeable. So claudin 24 potentiates also all calcium impermeable AMPA receptors in addition to GluA1, to the calcium permeable. Now, that this is not a desensitization effect is shown here. In the presence of desensitization, you see this potentiating effect. If you block desensitization with trichlormethoxide, it's still there. And if you take the ratio of the two, it's still there. So it's not an effect of the claudin on desensitization of the receptor, that's for sure. And it's also not an effect on the binding side, because the dose response curves are fairly similar or even identical, depending on the subunit that you're looking at. So there's no change in the EC50s, so the ligand binding site is not affected. IV curves are also not affected. And the question now was, is there a direct interaction between claudins and AMPA receptors, or is it indirect? Is there a protein in between that mediates this? And we looked at this by Fred analysis, by acceptor bleaching. So the acceptor is claudin, labeled with P-cherry, and the donor is the glutamate receptor labeled with EGFP. And if you bleach here the acceptor, you see that the fluorescence goes up for the donor because now it cannot transmit its energy anymore to the acceptor. And so you get a high FRET efficiency in this area, in this region of interest. If you pick another region and do not bleach, there will be no difference in the FRET efficiency. So in one case you have 24% FRET, in the other you have 0% FRET. So that proves that there is a direct interaction of the claudin with the receptor. And the next question is, are these actually expressed in the brain? Well, it had not been shown for these two claudins, 20 and 24. So we did this in collaboration with the Engelhardt group in Heidelberg 2 probes for Claudin 20, 2 for 24. As you can see, both are expressed in the cerebellum. Claudin 24 is also expressed in the hippocampus and in the olfactory bulb. So they are there where they could be working. And then since all the data I showed you so far, the functional data were encoded in oocytes, in the oocyte expression system, we tried to replicate this in hex cells. This is closer to a real neuron, obviously, so we did this in collaboration with Stephen Trinelli's lab in Atlanta at the Emory University. So here's the response of a glutamate receptor in hex cells. A non-active Claudin doesn't change that at all. Claudin 20 and 24 bring up the currents. And if you look at the steady state level, after desensitization, you see with the non-active Claudin, it's about the same as the wild type. In Claudin 20 and 24 co-expression, it goes way down. So the ratio of steady state to peak is changed. If you inhibit desensitization, you get these, instead of spikes, you get these long-lasting activations that last as long as you apply the agonist. And here again, you can see this potentiation, and you can see, if you look at the deactivation time curves, and this should be a red curve, I don't know whether you can see this, so it's right shifted for Claudins 20 and 24. So the right shift actually means that the deactivation is slowed down, so that explains why you get more currents. And so that concludes my talk, and the take-home lessons are certain Claudins are capable of potentiating AMPA receptors. Claudin 20 specific for GluA1, the calcium permeable variant. Claudin 20 in addition affects all calcium impermeable AMPA receptors. Kainate, NMD, and delta receptors we tested too, but we didn't see anything, except for maybe one editing variant of the kainate receptor. We're not sure about that. There's a direct interaction between Claudins and AMPA receptors. Claudins 20 and 24 both are expressed in the brain, as it should be if they are important physiologically. And you can see this in hex cells and analyze kinetics and show that the mechanism of this is slowing down of receptor deactivation. So the major conclusion then is not all members of the Claudin family, purported members of that family, actually may have a function as Claudin. Some may have either a completely different function or a little side job where they affect AMPA receptors. And with this, I just want to thank you for your attention, and this is my group, and this is the guy who did all the work, Simon here. Thank you.
Okay, just one question for our chairman.
No, not my question.
Oh,
Give the question to the audience.
Okay, sorry. Just one question. Yeah, please.
Do you think it's common binding site for Claudin and TARPs?
We have done competition experiments, and it doesn't seem to compete. But it's difficult to really find out because the TARP effect is much larger. So it's difficult to see whether it's additive or not.
Well, it's interesting, just specific, excuse me, just this is very important. Specificity actually for the calcium impairment. Because if they are touching each other and arginine is inside for this one, how the interaction is working?
You mean how the interaction
Between
with Claudin is working? Well,
specificity.
we don't really know where the interaction is. We believe it is inside the membrane through the transmembrane domains. But this is just our assumption. Now, if it would be the same as in the TARPs, then it might be the region just beyond the membrane. That has been shown for TARPs in the crystal structure at least to be the one that seems to interact. But I mean, no data for the Claudins exist. But the Claudins are homologous to the TARPs, and actually the guys who did the structure on the TARPs, they used the known structure of Claudins to model it after Claudins. Interestingly. It was Claudin 15 in that case, but yeah.
Okay,
I
thank
have
you.
time for more questions before I start.
Okay, yes.
Thank you for my talk.
Meanwhile, some more questions. Yeah, please.
What is the interaction between glutamate receptors and modulatory proteins depends on phospholipid composition?
It might well, but as I showed you, we saw this interaction in two completely different cell types, in oocytes and in head cells, which probably have very different lipid compositions. And we could show the very same potentiating effect in those systems. So while you're right, this may affect this effect or this modulatory effect, it is not preventing it as far as we can tell. But I mean, we haven't done the experiment to look at different types of lipid compositions here.